Very good morning, students. Today's topic is fermentation in industries. This video is in continuation with fermenter of my previous video. The term fermentation has been derived from the Latin word fervor, which means to boil, on basis of the boiling appearance of the action of yeast on extracts of fruits or malted grain during the production of alcoholic beverages. The boiling appearance has been due to the production of CO2 bubbles caused by the anaerobic catabolism of sugars present in the extract. However, today, fermentation is defined by biochemists as it is an energy generating process in which an organic compound is partially oxidized and partially reduced. Whereas microbiologists define fermentation as the process by which large quantities of cells are grown in a fermenter under controlled aerobic or anaerobic conditions with an aim of optimizing the growth of the organism or production of a target useful product. Industrially, fermentation may be performed in the following types, namely batch fermentation, continuous fermentation, aerobic fermentation, anaerobic fermentation, solid state fermentation, submerged fermentation, and surface fermentation. Batch fermentation is a closed culture system in which the microbial inoculum and fermentation medium and other components are added in specific amounts at the start of the fermentation process. These are not replenished once the fermentation has started. The product is recovered at the end of the process following cleaning and sterilization of the fermenter so that another batch can be set for fermentation process. In this fermentation, the microorganisms grow at a rapid rate in the initial stage in presence of excess nutrients, which is known as log phase. But as they multiply in large number, they use up the nutrients and secrete toxic metabolites, which results in retardation of the growth, leading to stationary phase and gradually death phase. This batch fermentation is similar to the one which we have studied about bacterial growth curve, which comprises of 4 phases of growth: lag phase, log phase, stationary phase, and death phase. Moving on to the second type of fermentation called continuous fermentation. In continuous fermentation, in contrast to batch fermentation, large volumes of fermentation media is taken once in fermenter in the beginning and the maximum yield is collected at the end. The continuous fermentations are those in which fresh fermentation media is added continuously by corresponding continuous withdrawal of a portion of the medium for recovery for fermented products. Here, media is being added whenever it is required by the microorganisms for the growth so that it maintains a constant log phase. There is no stationary phase or decline phase in case of continuous fermentation. These microbial cells never reach stationary phase, but because the end products do not accumulate as fresh nutrients are being added and the product is recovered in regular basis. The next type of fermentation is aerobic fermentation. In aerobic fermentation, the main feature is to provide aeration for the microorganisms, and the amount of air provided in aerobic fermentation is 60 times the medium volume. Therefore, bioreactors used for aerobic fermentations are usually stirred tank fermenters or airlift fermenters, where specialized parts like spargers or baffle plates are present so that air is supplied into the fermentation media for the growth of the aerobic type of microorganisms. Moving on to the next type of fermentation is anaerobic fermentation. In anaerobic fermentation, a provision for aeration is usually not needed, so they do not have any baffles or spargers, etc. But in some cases, aeration may be needed initially for inoculum build-up. In most cases, a mixing device is also unnecessary, but in some cases, Initial mixing is required. Once fermentation begins, the gases produced in the process generate sufficient mixing. The air present in the headspace of the fermenter is being replaced by CO2 or H2 or nitrogen or any suitable mixture of these gases, and it is particularly important for obligate anaerobes like Clostridium. So, apart from oxygen, oxygen is effectively replaced by CO2 or H hydrogen gas or nitrogen gas. The fermentation usually liberates CO2 and hydrogen, which are collected and used. CO2 for making dry ice and methanol, and for bubbling into freshly inoculated fermenters is being used. In case of actinogens and other gas utilizing bacteria, oxygen free sterile CO2 or other gases are bubbled into the medium, thus providing anaerobic environment for the growth of the suitable types of microorganisms. However, apart from what we have discussed like batch fermentation, continuous fermentation, aerobic, anaerobic fermentation, the most important types of fermentations which are used industrially are submerged fermentation, surface fermentation and solid state fermentation. Submerged liquid fermentations are traditionally used for the production of microbially derived enzymes. Submerged fermentation involves submersion of the microorganisms in an aqueous solution. So, obviously, the medium is in liquid form containing all the nutrients needed for the growth. Fermentation takes place in large vessels called fermenters with volumes of up to 1000 cubic meters. Batch fed and continuous fermentation process are common in case of submerged type of fermentations, and the parameters like temperature, pH, oxygen and carbon dioxide formation are measured and controlled to optimize the fermentation process. This is a diagrammatic representation of submerged type of fermentation, wherein we can see the fermenter is having a temperature control, an inlet for adding fresh medium, then acid base controlling units, then it has a rotor where it helps in growth of submerged type of micro fermentation, and an air inlet for entry of oxygen. In harvesting enzymes from the fermentation media, one must remove insoluble products, example microbial cells, and this is normally done by centrifugation. As most industrial enzymes are extracellular, secreted externally into the medium or the environment, they remain in the fermented broth after biomass has been removed. The enzymes in the remaining broth are then concentrated by evaporation, membrane filtration or crystallization depending on the intended application. If pure enzyme preparations are required, they are usually isolated by gel or ion exchange chromatography. So, these are the methods used for harvesting and recovery of the end products after submerged type of fermentation. Several types of submerged types of fermenters are known, and they may be grouped into several ways based on their shape or configuration, whether aerated or anaerobic type of fermenters, whether they are batch or continuous. The most commonly used type of fermenter is aerator stirrer tank batch fermenter. Moving on to the next type of fermentation of industrial importance is surface fermentation. In this surface fermentation technique, the microorganisms are cultivated on the surface of the liquid or solid substance or substrate. These techniques are very complicated and rarely used in industry. Aspergillus niger forms a mycelium layer on the liquid surface of the aluminum or stainless steel trays. These trays are stacked in fermentation rooms supplied with filtered air, which serves both to supply oxygen and to control the temperature of the fermenter. Surface fermentation is easy to control and implement. It needs no aeration or agitation of the fermentation broth. So, it needs no instrument for aeration and agitation, unlike submerged fermentation. The separation of citric acid from the mycelium is easy because of the microorganism is not dispersed into the medium. Only the temperature and humidity of the fermentation chamber need controlling. It can be used easily in small plants as well as in third world countries. With surface fermentation, the fermentation broth is concentrated due to high evaporation rate during fermentation. Thus, expenses and losses during recovery and purifications are low. However, surface fermentation has the following disadvantages. Building investment costs are high. Personal expenses are high in developing industrial countries with extremely high wages. Fermentation time is long, therefore, productivity is low. Submerged fermentation is favored over surface fermentation for the following reasons. Because low total investment cost is required for submerged fermentation, improved process control, reduced fermentation time, reduced floor space requirement, low labor cost, simpler operations, and easier maintenance of aseptic conditions on an industrial scale. Next, moving on to the third important type of fermentation is solid state fermentation. The origin of solid state fermentation can be tracked back to bread making in ancient Egypt. The solid state fermentations also include a number of well known microbial processes such as soil growth, composting, silage production, wood rotting, and mushroom cultivation. In addition, many familiar Western foods such as mold ripening cheese, bread, sausage, many foods of Asian origin, including miso, tempeh, and soya sauce, are produced by solid state fermentation. Beverages derived from solid state fermentation process include uh, Onjom in Indonesia, Shavo, Histing wine, and coiling sorghum from sorghum liquor in China, and sake in Japan. This is a diagrammatic representation of solid state fermentation, wherein the fermenter basically has a solid substratum with an air outlet and water cooling outlet, then perforated base, water cooling in, movement of water inside and outside of the device, and we also see a water reservoir and air filter and air inlet. Solid state fermentation is used for the production of bioproducts from microorganisms and conditions of low moisture content for growth. The medium used for solid state fermentation is usually a solid substratum like rice bran, wheat bran, grains, which require no processing. To optimize water activity requirements, importance for growth, it is necessary to take into account the water resorption properties of the solid substrate during the fermentation. In view of low water content, fewer problems due to contaminations are observed. The power requirements are lower than submerged fermentation, inadequate mixing, limitations of nutrient diffusion, metabolic heat accumulation, and ineffective process control renders solid state fermentation generally applicable for low value products with less monitoring and control. So, however, on the overall, solid state fermentation is of most preferable type of fermentation in case of industries. Here we can see some of the substrates used for solid state fermentation like sugarcane bagsy, tea waste, wheat bran, then sawdust, apple palm seed, then coconut oil cakes, etc. Advantages of solid state fermentations over submerged fermentation is higher volumetric productivity, usually simpler with low energy requirements, might be easier to meet aeration requirements, resembles the natural habitats of some fungi and bacteria, so easier to grow these organisms on solid state substratums. Then easier downstream processing, that is recovery of the product is easier here. The fungal hyphae are bathed in a liquid medium and do not run the risk of desiccation. Some more advantages are temperature control is typically not overall difficult, such that the organism is exposed to a constant temperature throughout its growth cycle. The availability of oxygen to the biomass can be controlled reasonably well at a particular level of saturation of the medium. The availability of the nutrients to the organisms can be controlled within relatively narrow limits if desired, though the feeding of nutrient solutions. pH control is relatively easy to provide. Next, moving on to stages of fermentation process. Now we have seen different types of fermentations which have been being used in industries. Let us see what are the different stages in a fermentation process in the industries. Industrial fermentation process requires two most important stages to complete product fermentation. They are upstream process fermentation and downstream processing. This is the diagrammatic representation of different stages of fermentation in case of industry. First one is upstream processing, wherein it involves inoculation of the media into the liquid broth or liquid media, inoculation of organisms into the liquid broth or liquid media, followed by fermentation process or fermentation bioprocess in a fermenter so that the product is produced in a desirable amount. After product is produced, then it moves on to the next stage of fermentation process known as downstream processing, which involves separation of cellular products, purification and concentration of the product. Upstream processing, it includes initial step of fermentation process. It can be divided into three steps. First, in order to reach upstream processing, we need the very first step is selection of the microorganism and optimization of the media, the selection of media in which these microorganisms can grow efficiently. Then preparation of the microorganisms for a industrial setting, and the desired microbes should be isolated, selected for the bioprocess. Then they should be cultured in a suitable growth medium. The second step is obtaining industrially suitable strains, preparation of media. Optimal conditions with the nutrients are supplied for the growth and multiplication of microorganisms in a bioreactor. The third step for upstream processing is optimization of growth conditions of the selected organisms, inoculation and running of the bioreactors until completion of the fermentation process. So this is the diagrammatic representation of upstream processing involving step 1, 2 and 3. Step 1, selection of microorganisms and media. Step 2 is industrially suitable strain development. Step 3 is optimization of growth conditions in a bioreactor. Moving on to the later part of industrial process called downstream processing, that is which is followed by fermentation. Downstream bioprocessing refers to several final steps of a bioprocess that involves product harvest. Downstream process starts when the product development is completed in a bioreactor. The step includes extraction of the product, purification of the product and packaging of the final bioproduct of the bioprocess. Downstream bioprocessing is also known as product recovery. Amino acids, antibiotics, organic acids, vitamins, vaccines are some of the bioproducts resulting from a bioprocess. The desirable quality of the bioproduct is achieved during the downstream process. Volatile products can be purified using a distillation process. Moreover, biomass separation can be done by centrifugation. Therefore, the stages involving separation, extraction, purification and polishing belong to downstream bioprocessing. Here we can see a flow chart of downstream processing, which involves if it is the product is intracellular product, then we need to disrupt the cell, then remove the insolubles, then isolate the product through adsorption, evaporation, extraction, precipitation or membrane filtration methods. After isolation, it is purified by chromatography, precipitation, membrane filtration methods, and the finished product can be obtained by crystallization, lyophilization, drying or sterilization, leading to the production of the final product. If it is extracellular product, so we don't need any cell disruption, it may directly move to the second step, that is removal of insolubles by centrifugation, flocculants, then filtration or sedimentation methods, followed by isolation, purification, finishing and final product. Thank you.